Yes, I've never seen that, and that was my first encounter. Maybe we just see it in movies and stuff, but that was the real life practical part of science I ever saw in my life. When I came to MLW, I was 14 years old. I really wasn't that hardworking student, but we met those doctors, Dr. Kondwani, for instance, and he had this outburst of motivation flowing around his face by his mind. Whatever he said was just encouraging.
When you do something that you have passion for, yeah, people will pay for it. It's 250,000. So how much do you think is new?
There was some passion deep in me, but what Kondwani did was that he just encouraged that passion. He increased the motivation, and I had more courage to pursue medicine.
I told him just now, right? I was once your age. Actually, I have always been the youngest. So looking at you, I look like myself. I was the same as you.
So I met Kondwani when we were at primary school, and Kondwani I think was about 11 years of age. He used to play on the team for the team that was our opponent team. Kondwani probably I would say was one of the youngest in their team, but much as he was younger, you would see him leading the team. You know, that was unique. You would see him, you know, being on top of things. He's there and he provides solutions, he provides direction. And I think that we saw it then when he was just, you know, a young boy. Even now we see that. You know, the school that he was selected to, like the secondary school, it was one of the, you know, top secondary schools, particularly in Zomba. So it's because he was doing well even in his studies. That's why he went to that school. And some of us went to another school. Not that we're not that good, but I think he did extremely well than us. When we were 12, you know, 11 years old, we didn't know what would, you know, become of us. And we didn't have that vision to say that one day we'll become scientists. My expectation was that probably he would end up being an engineer, being involved in construction and things like that.
I've had an observation for a number of years now, and it's been published, and if you go to the hospital, you actually find there are a lot of people in the wards that are HIV-infected individuals with lower respiratory tract infections, including TB and pneumonia. It was also the same 13 years ago when I started research. So that, to me, goes at the bottom of my heart, because I just don't feel right to see people dying from infections that we can easily cure. So the first objective was try to understand, is immunity in the lung similar to immunity in blood? A lot of studies in HIV were being done on blood, and we know that HIV-infected individuals have compromised immunity, specifically in the blood, but not a lot has been done on the lung. So that was the first question, objectives that I tried to address. So in science, what you normally do is you identify a problem or you observe something, and then you make a hypothesis. So a hypothesis is an assumption. In this case, I made the assumption that HIV-infected individuals have lower immunity in the lung specifically. That causes them to be more susceptible to respiratory infections. And after you generate that hypothesis, you now generate what we call specific objectives. So specific objectives are smaller questions aimed at trying to answer the bigger question. After generating these objectives, the next process you have to do is generate the methodology or design the experiments to try to understand these processes. What kind of samples can I use? And as well as what kind of participants can I have in the study? And this also requires a lot of reading and understanding what others have done in the field, and also generating new methods to investigate these smaller problems you've generated. So what we found from the first objectives was actually very interesting. We found that actually if you measure immunity in the lung compared to immunity in blood, if you took a blood sample, it's actually different, suggesting that if you just take blood samples and try to understand what happens in the lung, you might not get the full picture. So the idea of studying immunity in the lung was actually very insightful. And going forward, that's the approach that we've been using.
My expectation was that probably he would end up being an engineer, being involved in construction and things like that.
Kondwani, we made, I think that should have been 1999, just before we went to college. Kondwani did technical education and I did environmental sciences. So we used to be roommates and basically best friends as well. We were doing everything together. We were playing basketball for protecting pathos. I would look up to him on anything that I need, be it maybe I have problems school-wise, I have social problems. Yeah, so he was a very, very good friend, more or less like my brother. Those days we only had one university, so we were being given choices.
I chose to do accounting at the University of Malawi. Unfortunately, I wasn't selected to do accounting. I was selected to do Bachelor of Science in Technical Education.
Initially, we used to pay like 700 Malawi kwacha, and then they wanted to increase the fees to about 45,000. So the students striked, and it was the whole of the University of Malawi. That basically brought a lot of chaos at Polytechnic, College of Medicine, Chancellor College. So what the Chancellor decided to do was to close the university.
So building on from objective number 1, so in objective number 2 is immunity in the lung, specifically in the lung. When you compare between HIV infected and HIV uninfected individuals, is it different? So the assumption we made is that actually immunity in HIV infected individuals will be lower in the lung compared to HIV uninfected individuals, and that this might be the basis for the increased risk to lower respiratory tract infections in HIV-infected individuals.
So the outer layer, actually the most.
So in order to answer the questions that we set up, we need to collect samples. So we decided to collect blood sample and the lung sample. The volunteers, and these are volunteers, they are not coerced or forced to go into these studies.
Once they come here, we explain everything. Because sometimes just to explain the procedure, it is not easy. And most of them, they're afraid. They think maybe they'll die. But we try as much as possible to explain everything so that they must know what complications can arise after doing bronchoscopies. So they have time to ask. Others, when they go home, after reading the information, others, they call me. They say, "Sister, can you explain again?" So we explain the procedure again.
And once they sign and say they are okay in joining the study, they are also told that they can withdraw from the study at any point, even during the procedure. But to get the lung sample, there's a technique that we use. It's called bronchoscopy and bronchoalveolar lavage.
Once the patient is ready and we have checked that oxygen saturation and the doctor is ready, so she intubates the bronchoscopy in the nose. And once they reach the throat, we give lignocaine 2%. Lignocaine jelly is an anesthetic which we use so that the patients should not feel pain. Once they reach the carina, then the doctor positions the scope to the right side where we take our samples. So we push 200 ml of warm saline.
That fluid then is pumped into the lung, and we wash part of an area of the lung, and we take it back into the scope. So in that container, you now have the fluid saline plus whatever has been washed from the lung. And that is now taken into the lab for the next step, which is actually sample processing. So in response to objective 2, what we found was that actually immunity in the lung in HIV-infected individuals was actually lower than HIV uninfected individuals, which is the assumption we had made. And this was actually very exciting because the difference between these groups was observable in the lung more clearly than it was in blood. As well, this fits very well with our area observation that immunity in the lung is different from immunity in blood.
So we were sent home indefinitely, everyone. So we decided to do something with the time that we had. So we came to MLW, and I still remember by then the director was Marco Molyneux. We met him and we asked him to do something voluntarily just to pass time, to learn what these guys are doing here by those days. So we brought the CVs, and then he looked at us. He wrote on I remember, on one of the copies of the CV that voluntary, if necessary, payment if possible. So we went home, and then a day after, they called us for interviews. To be honest, I had no clue what these guys were doing here. Basically, Kondwani was escorting me to drop my CV because we knew that they are doing lab-based stuff here. And similarly, I was supposed to escort him and drop a CV somewhere where actually they're doing something that is similar to what he was doing at school. Basically, we knew so little about sciences, and the people that we would look up to were people who mostly doing marketing studies, selling cars in town and all those things. So at some point, we really thought that maybe when we finish, we should do a marketing professional qualification.
So following on from objective 2, I went on to objective number 3. So objective number 3 takes into account that HIV-infected individuals are now put on antiretroviral therapy immediately as soon as they're found to be HIV positive. So what we're trying to address is, does this antiretroviral therapy reconstitute or improve immunity in the lung in HIV-infected individuals? This is very important because there's also observations that have shown or suggested that HIV-infected individuals on treatment are still more susceptible to lower respiratory tract infections compared to HIV-uninfected individuals. So in this case, we get samples from Queen Elizabeth Central Hospital. So we have a blood sample for that individual, and we have a lung sample for the same individual. And we go through a process of centrifugation. We call it density centrifugation. For the blood samples, we add a chemical called lymphoprep. So that also helps in separating red blood cells from white blood cells. What happens, because you have been spinning them on high speed, the cells, because they'll be much denser than most of the things that are in that sample, will sink to the bottom of the tube. And then we take a layer of white blood cells from that. Those are your actual cells. So we put nutrients, what we call a medium. So the media usually represents the similar conditions that you would find the cells in the body, so that the cells should have energy and should be able to survive. And after that, you now have 2 samples which contain cells, and these cells are white blood cells from either the lung or from blood. And then we do experiments using those cells. With objective 3, what we found is that HIV-infected individuals that were on treatment still had lower immunity against lower respiratory tract infections, but it was higher than what you would find in HIV-infected individuals that are not on treatment. And that also reflects the observation in the hospital that the risk to lower respiratory tract infections in HIV-infected individuals that are on treatment is actually a bit lower than if you compare that to HIV-infected individuals that are not on treatment. And because of that, we are really very much interested in trying to understand this a bit further, to go a bit in depth into this. And we designed a 4th objective.
I'd never met him, but one day our boss, Professor Molyneux, came into the lab and said there were 2 students from the Polytechnic who wanted to do a project with my group. And we were very busy, we had no time for students, so I said no, we were too busy. And the next day the boss came back again and said the 2 students were really quite keen, and the Polytechnic was on strike, so they had nothing to do and they didn't want to be paid, but they really wanted to do a project. And I said, definitely, I've got no time. And my boss went out and said to Kondwani, you start on Monday. And so Monday came and Kondwani turned up with his friend Kuzwayo Jerry.
So we came here and then we were given a tour to see what's in here. Actually, to be honest, quite impressive because we didn't have that at Polytechnic.
They started to look at lung cells. I found very quickly that they applied themselves in a very determined way to the project and quickly found some problems with the method that I was suggesting.
Mind you, I was at a government school where everyone basically does things at their own time, whereas here you had to stick to deadlines, and our boss actually was challenging us to do things sometimes that we've never done before. So that challenge itself, I think, was quite stimulating.
So that small piece of work that started with 2 students coming from the Polytechnic and applying themselves in a determined way has led to many tens of thousands of people being studied and opening up a whole new field within Malawian health.
This was the first time when we actually published in internationally recognized journals, which perhaps if we were at Polytechnic we would never do that. And we did that actually before we finished our first degrees. So yeah, that was quite impressive.
So when Kondwani and Kuzwayo started on that Monday morning, they were given an incredibly dull task. So they had several hundreds of slides of lung cells, and they had to sit in a little dark room about twice the size of a toilet and take pictures of lung cells through the microscope. And it got a lot more boring after that, because when they now had several thousands of pictures, they had to use a computer program to count the amount of blackness in each slide. And this went on for weeks and weeks and weeks.
So now the objective is trying to understand why is it that HIV-infected individuals on antiretroviral therapy still have a higher risk to lower respiratory tract infections compared to HIV-uninfected individuals. Specifically, why is the immunity in the lung still remaining lower and doesn't reach to the levels of an HIV-uninfected individuals in these HIV-infected individuals that are on treatment? So once we take the samples that have been processed, so a lung sample and a blood sample, what we do is then have to count how many cells do we have in each sample. So we take the cells from the sample, just take a small fraction. I want 1 million cells per mil in one lung sample that you're looking at, as well as 1 million cells per mil in your blood sample, so that they should be at least of similar numbers. You take your lung sample and you take your blood sample and you stimulate. To stimulate basically means you give these cells some parts of HIV called peptides, because what happens is when your cell sees HIV or parts of HIV, it responds. Now that is what we call an immune response. So a process of stimulation now started doing an experiment, and you incubate them in a cabinet that has similar temperature to your body, which is 37 degrees. So after 6 hours, if the cells have seen HIV before, they respond by producing proteins. The level of proteins will be different between individuals depending on the state of their immune response. And then we do a next step, which is called staining. In the staining process, what happens is you take antibodies that have a fluorochrome. They're antibodies against the specific proteins that your cells produce. If those proteins are available, the antibodies will bind. If they're not available, they will not bind to anything. Then you do a washing step. It removes antibodies that have not bound to anything. and we take those samples that we have stained onto a machine called a flow cytometer. It measures light. When there's light, it excites the fluorochrome and it emits a particular light. So it can be different colors of light: blue, red, green. If your cell is expressing protein A or has protein A and protein B, they'll be on different colors on the flow cytometer. And using different combinations of colors, we now are able to identify a specific cell and what protein it is producing. We have to produce graphs because it's massive information you're trying to look at. Then you do statistical tests on them. Using a statistic, you'll be able to see if the differences are real or they're just by chance, that you shouldn't make a subjective observation. Then you can compare that between individuals or between groups. And from there, that's when we get our answer to the question that we're trying to address in the first place.
So their first job was extremely dull, but they were very determined. And then when they got through that first job, they actually went back to college. And then when they came back after their term, they finally got to unpick the answers as to what was causing the smoke in the cells of Malawians.
So initially, I never wanted to be a scientist, and I never knew what being a scientist was all about. But when I first came here in 2003 at MOW, I was introduced in the field of science. And one thing that caught my attention is the idea that you can observe something, make an assumption about it, set up a process, try to identify if your assumption is correct or wrong, and then at the end of it, you have an answer. And that has actually even shaped the way my thinking process is, even at the time, at that point in time, in so many different things. And I always tell people to always challenge your assumptions.
My impression is that their interest has grown as the complexity of the questions that they have addressed have grown. And so both of them, as it turns out, have gone away from studying smoke, and both are interested in infection, which is much more complicated because it's the interaction of a living organism with the human being.
The most immunogenic part of this. Yeah. So while I was at MOW doing my internship, but I had to decide which field I liked the most. I was really interested in understanding immunity, and I got a scholarship to go do a master's in immunology at the University of Liverpool. I was doing a Bachelor of Science in Technical Education. There is no connection with immunology. I went to a class where people had a biology and immunology background. So the first objective was try to understand, is immunity in the lung similar to immunity in blood? So I was set to do research in this particular area.
I've worked with him as somebody who helped him, who advised him on how to get funding, and then he worked on the projects with us. But I did contribute to part of his PhD training.
We recognize him as a good scientist that we have here in Malawi, and he will soon be recognized worldwide that is a fantastic scientist.
So he's no longer junior to me because each one of us is an independent researcher. He may be junior in the sense that we're different in age probably, but in terms of the work we do, it's very, very complementary.
What I really want to do next is now try to find what is it that we can do about that. So trying to identify ways and means, therapeutic way, of either populating the anti-HIV immunity in the lung or finding drugs that can be given in addition to ARTs in order to reduce or eliminate HIV in the lung. This is from mouse, but it binds to a human protein.
And if you look at Kondwani at this point in his career, he has had fantastic publications in very high quality journals, and that speaks for itself.
Because as being a Malawian, somebody else had to inspire me to see that it's possible for everybody else. Now I'm more interested in getting more people to be like me. So to be wanting to have the passion to do science, at the same time help address the problems that are facing in terms of health in our population.
I mean, he'll go a long way. I mean, I would not be surprised. And certainly in academia, you know, a chair or professor in an academic institution. And I think that's something that he will achieve at a very early age.
What Kondwani did was that he just encouraged that passion. I had more courage to pursue medicine. My name is Makion Chitsego. I'm a student under Bachelor of Medicine and Surgery, MBBS, but we're in our first year, so it's called MBBS 0. As opposed to Kondwani, who is an immunologist, I'm interested in the heart, so I want to specialize in cardiology.